Distinguished colleagues, esteemed participants, I am very happy to be with you in this wonderful academic environment and at this highly valuable scientific congress. I would like to express my gratitude to the Congress Organizing Committee for their efforts. Today, I will talk to you about the probiotics found in our gastrointestinal system, the prebiotics that interact with them, and especially the roles that short-chain fatty acids produced as a result of this interaction play in our health. In light of the latest scientific data, I will discuss both the mechanism of this and the role of the microbiota, particularly its relationship with short-chain fatty acid production in long-term treatment strategies related to our health. There is a very strong ecosystem in our gastrointestinal system. Here, we coexist with trillions of living organisms, most of which are bacteria. We've started to realize that these are just the tip of the iceberg as we understand and grasp the mechanisms involved. In other words, the interaction between the endothelial cells in our gastrointestinal system and various specialized cells among them, such as Paneth cells and enteroendocrine cells, and this ecosystem and its products plays a role in regulating our immune system. This regulation primarily affects intestinal health and subsequently is closely linked to both systemic diseases and our overall health status. In other words, we are only seeing the tip of the iceberg right now. But with each passing day, science continues to advance, gradually discovering just how vast the part beneath the iceberg truly is. Speaking of living organisms, it's not just our gastrointestinal system. Starting from the oral cavity all the way to the skin, there are actually a great number of bacteria present in many areas of our body. When we specifically look at the gastrointestinal system, we live together with a large group of bacteria called probiotics, whose numbers and diversity increase from the stomach to the colon. Their concentrations range from 10 to the power of 2 up to 10 to the power of 12, while their diversity exceeds 1,000. We have prebiotics that interact with these diverse bacteria and serve as their food source. These prebiotics are made up of somewhat more complex carbohydrates that are not affected by digestion in the digestive system. Here, as we can see on the list, there are many indigestible complex carbohydrates called prebiotics, such as inulin, beta-glucan, ephos, GOS, arabinoxylan, and pectin, that are not affected by digestion in the digestive system, but are processed by the bacteria in this ecosystem. The effects of these complex carbohydrates are being understood more and more each day. Their suppressive effects on chronic inflammation, their ability to promote the growth of beneficial bacteria here, and the production of short-chain fatty acids through interaction with these bacteria not only contribute to the movements of our gastrointestinal system, they do not just contribute to peristalsis. At the same time, they prevent this area from being exposed to various toxins. They also prevent unwanted bacteria from colonizing here. And in addition to these, they play a role in the pathophysiology of many chronic diseases in our body, especially diabetes. If we look a little more deeply, as probiotics process prebiotics, that is, microbiota accessible carbohydrates, our short-chain fatty acids, SCFAs, are formed. As examples of these, we can first mention acetate, propionate, and butyrate. And these are the three short-chain fatty acids with the most effective functions in our gastrointestinal system. If we take a look at the general picture, fatty acids with 2 to 6 carbons are considered our short-chain fatty acids. The list includes various fatty acids such as acetic acid, propionic acid, butyric acid, and isobutyric acid. In particular, this trio, acetic acid, propionic acid, and butyric acid, stands out because they nourish colonocytes and contribute to their energy metabolism, and as a result, the health of our gastrointestinal system is especially positively affected by their presence. If we take a closer look at these again, we see that they are formed through different pathways. here is acetate, here is butyrate, and here is propionate. And as you can see here, there are many different mechanisms by which these are formed. While the classic glycolysis pathway that we know goes from acetyl-CoA through the acetoacetyl-CoA pathway all the way to butyrate, we also know that propionate is produced by gut bacteria using these prebiotics through pathways such as the acrylate pathway and the succinate pathway. When we look at which bacteria specifically produce acetate, butyrate, or propionate, we know that certain bacteria such as Akkermansia, Mucinifera, Bacteroides species, Bifidobacterium species, and Prevotella species produce acetate, and that some bacteria like Coprococcus, Eubacterium, Faecalibacterium, or Roseburia produce butyrate. When it comes to those that produce propionate, it is known that gut-friendly bacteria such as Megasphaera, Bacteroides, and Salmonella produce these short-chain fatty acids through 3 different pathways. If we take a closer look at the health benefits of these prebiotics, such as inulin, FOS, GOS, pectin, and beta-glucan, for example, inulin. Here, we see the bacteria that process inulin, in other words, the bacteria that metabolize it and produce short-chain fatty acids. And as a result of this process, we know that acetate and butyrate levels increase, which contributes to digestive system health, supports mineral absorption, and optimizes the immune system to an ideal level. Similarly, as prebiotics, FOS, fructooligosaccharides, contribute to satiety by increasing the production of acetate and butyrate, help regulate blood sugar levels, and are beneficial for the overall health of the digestive system. When we look at galacto-oligosaccharides, we see that they contribute to our overall gastrointestinal health by increasing the production of propionate and butyrate, and also by enhancing calcium absorption through the immune system. When we come to type 2 resistant starch, you can see here, based on various studies and recent discoveries, that it improves insulin sensitivity by increasing the production of acetate and butyrate, contributes to the feeling of satiety and taste, and through all these effects strengthens the health of our gastrointestinal system. When we look at the years of these studies, we will see that all of these discoveries have actually been made in the last 20 to 25 years. Let's take a much closer look together at precisely how the living organisms within this intricate ecosystem demonstrate their fundamental mechanisms of action. These complex carbohydrates, which we call functional oligosaccharides and which serve as food for our gut bacteria, are processed by bacteria such as Lactobacillus and Bifidobacterium and are primarily converted into short-chain fatty acids, mainly acetate and lactate. Then, through further conversion by bacteria such as Roseburia, Faecalibacterium, and Legionella, they are transformed into other short-chain fatty acids like propionate and butyrate and begin to exert their effects on the epithelial cells of the gastrointestinal tract. Again, while these beneficial bacteria indirectly prevent the proliferation of harmful pathogens here, these remarkable functional oligosaccharides also exert their significant effect by contributing to the essential lectin production and directly inhibiting these truly harmful bacteria, thereby effectively limiting their undesirable presence in that specific environment. We also see how prebiotics, especially through Lactobacillus, Bifidobacterium, and Faecalibacterium species, support the tight junction proteins that hold together the cells in our gastrointestinal system. In particular, here they have a direct and significant effect on maintaining the structural integrity of crucial tight junctions, such as the well-known proteins ZO1, occludin, and claudin, which collectively ensure the robust strength of these vital cellular connections and effectively prevent problematic and undesirable conditions like a leaky gut. Additionally, by diligently producing beneficial short-chain fatty acids, they also indirectly contribute to the overall health and optimal functioning of these essential junctions. Again, by affecting goblet cells through mucus secretion, the release of certain antimicrobial peptides helps maintain beneficial bacteria in the gastrointestinal system at optimal levels. This is how the mechanism is regulated. If we take a closer look, especially the so-called microbiota accessible carbohydrates, or MACs, contribute to tight junction health by affecting different bacterial species such as Akkermansia and bifidobacteria, as we mentioned earlier, or by stimulating these cells to increase the synthesis of GLP-1 and GLP-2. This helps organize and maintain the physiological integrity of that area at the most optimal level. Again, the short-chain fatty acids produced through interactions with bacteria directly contribute to tight junction health. Indirectly, acetate also supports the health of this area through the production of interleukin-18 as a result of NALP3 activation. Again, bacteria such as those producing acetate, butyrate, and propionate are used especially as fuel for our cells in the intestinal or colonic regions. Whether by directly entering the intricate TCA cycle, also known as the Krebs cycle, or by first undergoing the process of beta oxidation, these relatively short-chain fatty acids directly and significantly contribute to providing essential energy for our various cells here within the body. To comprehensively summarize these significant effects, the notable increase in mucus secretion observed here, the crucial transformation of dendritic cells into T helper cells via GPR109A, and the subsequent secretion of interleukin-18 all demonstrably exert their profound impact in effectively regulating the intricate activity of our immune system's various cells. Again, their significant contribution to the crucial secretion of essential tight junction proteins, which we have just mentioned, and finally, the comprehensive production of vital proteins such as GLP-1 and PYY via the specific GPR43 receptor, all collectively help to effectively maintain optimal intestinal health and overall physiological balance within the body. This remarkable achievement is primarily brought about through the intricate and beneficial interaction between the beneficial bacteria and the prebiotics present here. Let's take a closer look at precisely how these important mechanisms work, especially at the cellular receptor level. Short-chain fatty acids exert their primary effects mainly through histone deacetylase inhibition or by activating specific G-protein-coupled receptors. Histone deacetylase enzymes and G-protein-coupled receptors contribute significantly to our immune system, our antioxidant system, glucose and lipid metabolism, as well as the inhibition of processes potentially leading to cancer by increasing the expression of GLP-1 receptors or nuclear factor 2 receptors. It exerts these beneficial effects by activating some of them and inhibiting others. Again, when we take a closer look at this ecosystem, we know that NLRP6 receptors and NOD2 receptors are particularly effective through interleukin-18 or through the impact of T helper 17 cells on Paneth cells. Similarly, short-chain fatty acids exert their effects by acting on G-protein-coupled receptors, thereby influencing the PPR-gamma signaling pathway within the cell, increasing the expression of GLP-1 and PYY, and enhancing the secretion of Treg cells of cytokines such as interleukin-10 and transforming growth factor beta, which, as you know, are our anti-inflammatory cytokines. Here we can see how these processes within the gastrointestinal system affect our overall health through immunology, specifically via the immune system. We can see here the mechanism by which dendritic cells, by transforming into T helper 1, T helper 17, or beta cells, and through the mediators they secrete or the cytokines produced by the newly formed cells, such as secretory immunoglobulin A, interleukin-17, and interleukin-1 beta, play a role in maintaining the overall health of our immune system or in the pathway leading to diseases. If we take a closer look at the relationship between short-chain fatty acids produced in this ecosystem and diseases, we can clearly see the effects of these short-chain fatty acids, especially in a very recent review article. Our first disease is diabetes. In a randomized clinical trial conducted on diabetes, it was observed that short-chain fatty acids were decreased in patients with type 2 diabetes, and that with high fiber intake and prebiotic consumption, the levels of short-chain fatty acids increased, as did the populations of bacteria that produce these short-chain fatty acids. Again, in a meta-analysis related to diabetes, it was found that increasing butyrate and propionate, as well as increasing bifidobacteria, occurred as a result of adding fiber to the diet. When we look at inflammatory bowel diseases, a study conducted on a series of 313 patients found that there was a decrease in bacteria responsible for the conversion to acetate and butyrate in this patient group, a decrease in propionate levels, and an increase in undesirable pathogens. Again, in a study comparing 127 patients with 87 healthy individuals, it was found that in these patients with IBD, there was a decrease in butyrate-producing bacteria and a reduction in short-chain fatty acids. Similarly, in a group comparing 10 patients with Crohn's disease and 10 healthy individuals, it was found that bacteria-producing SCFAs had decreased, and in particular, bacterial species such as Roseburia, Ruminococcaceae, Clostridium, and Bacteroides had declined. Similarly, in 3 separate studies conducted on non-alcoholic fatty liver disease, especially in the initial part of these regions, that is, when 14 patients with non-alcoholic fatty liver disease and 18 patients with steatohepatitis were compared with 27 healthy controls, it was found that there was an increase in the production of short-chain fatty acids in these patients, and an increase in the bacteria that produced them was detected. Similarly, many studies like this, especially those conducted on neurodegenerative diseases such as Parkinson's and Alzheimer's, have consistently demonstrated a relationship in these regions regarding the bacteria that produce short-chain fatty acids and the quantity of these bacteria. When we look at the roles of short-chain fatty acids, especially butyric acid, in these areas, through a literature review based on a specific example, we see that the number of studies has increased significantly in recent years. There are 105,000 studies, and when we search PubMed, we can see the growing focus on these topics in recent years. Butyric acid is a topic that has been studied since the time of Louis Pasteur, specifically regarding which bacteria produce it and how. And when the contribution of butyric acid to our health was investigated, science discovered that these short-chain fatty acids are regulated especially by the intestinal microbiota in our colonic region, and that various groups of bacteria need to be present there to make this happen. The presence of Eubacterium, Roseburia, Faecalibacterium, and Coprococcus, especially as we just mentioned, in the colonic region is essential. Their presence enables the metabolism there to function properly, supports protective functions, and helps prevent various problematic conditions related to gastrointestinal diseases. When we look specifically at this area, the healthy execution of absorption or secretion processes by the intestinal mucosa is made possible by the presence of butyric acid. Again, the anti-inflammatory properties of butyric acid arise from its inhibition of the NF-κB pathway. To achieve this, they inhibit the gene regions responsible for secreting our pro-inflammatory cytokines, such as TNF-alpha and interleukin-1 beta, and by exerting their effects in this way, they suppress reactive oxygen species. That is, they have antioxidant properties. Additionally, by increasing glutathione levels here, they explain the protective mechanism, especially in inflammatory bowel diseases, through these positive effects. And if we look at their mechanisms of action, we had especially mentioned G protein-coupled receptors. To this, we can add hydroxycarboxylic acid receptors to the free fatty acid receptors. It has also been definitively discovered that inhibitions via histone deacetylase, which are crucial enzymes, effectively regulate cellular homeostasis by profoundly affecting the intricate energy mechanism here, and at the same time, due to their significant and pervasive effects on transcriptional activity, they powerfully inhibit the complex process leading to the development of cancer. In other words, our short-chain fatty acids exert their effects through these receptors across a very wide spectrum, ranging from various allergic diseases to different types of cancer, in addition to disorders where immune functions and inflammatory processes are adversely affected, such as diabetes and obesity. Many studies like this one have examined the effects of butyric acid, especially in different disease groups. Its positive effects have been identified in irritable bowel syndrome. Again, both in the intestinal region and extraintestinal regions, the health benefits of butyric acid have been demonstrated due to its effects on various areas such as insulin sensitivity, cholesterol synthesis, and neurogenesis. Another example is the EPIC study, an ethical study examining the relationship between cancer and nutrition. Research has shown, with supporting evidence, how butyric acid is necessary under physiological conditions in colorectal cancers and also necessary for inhibition under pathological conditions such as cancer. Especially regarding its significant effects on the immune system and systemic inflammation, it is repeatedly emphasized that butyric acid is an exemplary molecule for addressing issues in this crucial area. These points are being highlighted. Finally, there is a recent study conducted on patients with polycystic ovary syndrome. It thoroughly explains how butyric acid effectively exerts a significant positive effect, specifically through its profound impact on various inflammatory processes within the body. In the obese PCOS group, it was specifically found that the levels of butyric acid were particularly and notably low, and the supplementation with exogenous butyric acid was clearly shown to significantly improve the overall ovarian functions in these specific PCOS mice, demonstrating a beneficial effect on their reproductive health. Together with my esteemed colleagues Amin and Maryam, in the comprehensive book we meticulously wrote, we earnestly aimed to draw significant attention to this crucial topic and contribute meaningfully by thoroughly emphasizing precisely how these short-chain fatty acids profoundly affect our entire, intricate overall health system, but especially their substantial impact on inflammatory bowel diseases, and by presenting compelling, detailed data from our extensive research that strongly supports these vital points. Thank you all very much for your kind attention and for listening so attentively. I hope to see you at another Congress. I would especially like to thank the organizing committee. I wish you all a great Congress.